glucose 6-phosphatase that is also embedded in the membrane, but whose active site is facing into the lumen. And this hydrolysis generates glucose and phosphate, and both of those metabolites are transported out. And the glucose that's transported out is either shuttled directly from here to here without really being free in the cytosol. I suppose if it became free in the cytosol and it was broken down to G6P, or it was phosphorylated to G6P, then it could come back in. That wouldn't be very effective because you'd be using up ATP in order to run this this cycle here. So although this slide doesn't show it very well, there must be a way for this glucose to get through here and not become free in the cytosol, but just simply go through from the ER lumen to this, what's called GLUT2, this receptor in the outer membrane of the cell that can get the glucose out to the bloodstream. So that's kind of a neat little process by which liver cells can circumvent the problem that they would have of using up their own glucose rather than exporting it. And they make use of another compartment that they have in the cell. Questions about that? Okay. So glycogen synthesis. This slide is not really about glycogen synthesis yet, but it's just here to remind you that when we talked about how galactose was mobilized to go into the pathway to break down for glycolysis, we learned about these sugar nucleotide molecules, in particular UDP glucose is a sugar nucleotide that's used in this pathway. And so you can go back and look at that little pathway that we mentioned when we talked about how to use galactose. The UMP portion of this, so this is glucose linked through the C1, right, to 2 phosphates, right? But we would have here basically what amounts to glucose 1-phosphate in UMP, right? So UMP can be a good leaving group, if you imagine this leaving, to generate glucose 1-phosphate. So this is how sugar molecules are synthesized from a sugar phosphate and NTP. Here's the sugar nucleotide. So glycogen synthesis actually starts with UDP reaction with glucose 1-phosphate to generate UDP glucose. That's that enzyme here. And then UDP glucose is used by this enzyme glycogen synthase. And here's glycogen synthase. So here's glycogen. And now we're adding to glycogen. Right? So we're in an energy-rich state, presumably, and the cell has so much energy that it's able to run gluconeogenesis and now make glycogen. So here's UDP glucose, and here's the non-reducing end of a glycogen chain with some number of residues. Notice that N has to be greater than 4, so there's more steps that I'll talk about in a moment. But in this reaction of glycogen synthase, UDP is split off. So here's UDP. And then this glucose molecule is attached onto the elongating glycogen. So what we have here is, this is the nucleophile, this is the electrophilic center, here's the leaving group. This bond is going to be broken right there to make UDP. Okay, so this is a heavily regulated enzyme. So you think of the 2 central enzymes are, one is phosphorylase to do phosphorylasis of glycogen to generate G1P, and the other one is the synthase reaction. Right? And so those are the workhorses. The rest of the enzymes, like the mutase or the branching and debranching enzymes, are there because we want to efficiently build a particle that's branched. So that's more efficient for the storage and more soluble, or we're just connecting to other parts of metabolism. So just like we had a debranching enzyme, we have a branching enzyme. This is not shown to depict any sort of mechanism here, but the idea is simply to show that if you can, this sugar here is linked to this, and it's also linked to here. So now we have branched sugar here, which is going both ways on 1,6 and then the 1,4 linkages this way. So what's basically happened is this amount here, all of this stuff has transferred onto this sugar. And now we have a branch point, which is a distinctive linkage as compared to, the 1,4 linkages through here. Now we have a 1,6 linkage here. Okay. So this will have two effects. It makes the whole particle more soluble, and it also increases the number of non-reducing ends. And that's important because that's the way you add more glucoses. So the more branched it is, the more individual non-reducing ends you have, and the faster that you can build up your glycogen particle, because you can build up on all those non-reducing ends all at once. Okay. So we need the branching enzyme. And so what we haven't talked about yet is how we start the glycogen particle. So glycogen synthase requires a certain length on the end, and then we have this branching enzyme that when we have long lengths, it will transfer some of the pieces around, so it will make the branches, and you get to the end of the particle. But what about the very beginning of the particle? So the very beginning of the particle relies on this enzyme here. It's called glycogen. And the way glycogen works is it has a tyrosine in its active site, and it's using the same substrate as glycogen synthase is, like UDP glucose. It attacks at C1, releases UDP in the same way, and then it has here's the enzyme with the tyrosine with one glucose monomer attached and a free C4, which can then, by the activity of this same enzyme, attack another UDP glucose, release another UDP, and now you've got two. And then it does this up until its chain length of about seven or so glucoses, something like that. And so at that point, glycogen synthase can come along and add more, and then eventually the branching enzyme can come along and branch it. And so all this buildup of cabbage of glucoses builds up around this central particle. But if you were to take a glycogen molecule and peel away all the stuff and get to the very core of it, there would be one molecule of this little protein hanging out in there. So every glycogen particle has one molecule of this protein in the middle of it. Right. So it's kind of interesting that this enzyme, you can kind of see, well, how it could do the first reaction, because it has a tyrosine, has a UDP glucose on it. It just has to juxtapose the UDP glucose with its tyrosine in the active site, maybe have some catalytic residues around, and then it could do this reaction. Right? But then it does another one, and another one, and another one. The same enzyme is doing all that. And this chain is starting to get, 2, 3, 4, 5 residues long. So if it's 2, 3, 4, 5 residues long, isn't it kind of coming off the surface of the particle? So how does it keep on doing it? I don't have an answer to that. But it would be interesting to look into how that activity keeps on working that way. Right? 'Cause it's not that another enzyme comes along, because we know that there's not multiple covalently linked protein molecules inside the particle. So it seems a little counterintuitive in terms of just how at least I like to think about enzymes. And I haven't looked into it in depth, so I don't really have a good explanation for why that works. So you guys understand at least why I'm confused about it? Maybe one of you guys is not confused, you can explain it. Yeah?
So just to clarify, the glycogen just creates like the first thing, and then we have glycogen synthase and the branching enzyme to create all the rest of the tiers?
Yeah, that's right.
Okay.
Yeah. Yeah.
So like you said the first 4 molecules or so are just by the glycogen?
I think it's more like 6 or 7 or so.
So that's confusing because it's happening like out of the active site?
Yeah, or I just imagine that it would be happening out of the active site. So maybe the thing curls around somehow, something's happening. But it's a little bit inconsistent with my notion of how, because it's covalently stuck on one end. And that's kind of where the active site is, or else you wouldn't get the first reaction, right? Does that make sense? I mean, this is the first reaction here. Here's a tyrosine. I mean, it's in the active site here somewhere. Now this glucose is in the active site. Now you've got to react on this end of it, and then this end, and then. Right? I'm confused. All right. So let me just, I'll just summarize all that. So the breakdown